All right, so the problem here is that we're looking at how plants transport water, right? There's lots of different ways that they could do it. There's really only one way that they do, in fact, transport water, right? And we talked about that yesterday. We talked about that being an osmotic pulling action from above, okay? As water evaporates from the surface of the leaf, okay? Salts and minerals get left behind, which creates an osmotic pressure because water wants to move across membranes to balance salt, so it moves into the spongy layer of the leaf, okay, to balance that salt concentration. But because water is a polar molecule, it is hydrogen bonded to both other water molecules and the inside of the tubes, the xylem that we learned about yesterday, okay, the xylem tubes, okay, it's hydrogen bonded to those. So as one water molecule moves up, it pulls on all the others below it in that tube, pulling them up the plant, okay? So it's actually osmosis, which is passive and requires no energy from the plant, that is helping to move water up the plant, okay? There's no pumps in the roots, okay? There's nothing in the stem that's lifting it up, okay, or anything like that, right? So since we are fairly certain that the source of this force is in the leaf itself, we need to confirm that, okay? So now we're talking about the design here. Okay, so the problem is basically, how does it work? Okay, you just have to rephrase that in the form of a question. How does a plant transport water? Okay, but now that we're talking about the design, okay, now we have to look at this. If we're fairly certain the leaf is the source of this osmotic pulling force, we want to eliminate any other parts of the plant that could possibly have a role to play in moving water. Okay? In other words, we're manipulating the plant. We're changing it. We're taking some parts of it off so that only the leaf is left. That way, if water moves, there's only one place it can come from. Okay? Only one place that force can come from, and that's what's left of the plant. All right, now, our responding variable here, okay, well, actually variables, are the things that we are recording or observing about the plant, okay? There's essentially two things that we're making observations about. One of them is qualitative, okay? And you've taken two pictures of it now to do with the qualitative part. And a quantitative part, the one thing we're actually measuring, the stuff we're gonna pour out tomorrow when this is all done, okay? You find a way to put that in your own words and explain how and why it's changing. Okay, and then controlled variables. These are going to have to come up with, but this is the tip that I'll give you. Anything that can affect the rate at which a plant carries out photosynthesis or transports water will need to be controlled throughout the experiment as best we can. Okay? So think about what kinds of things might affect the growth of a plant that might be kept the same in this room on that windowsill, okay, for 2 days. Okay? Make sure you explain each one of those three controlled variables by telling me this is the effect that this has on transport, photosynthesis, growth, whatever. Okay? This is why it has to be controlled. Okay. For our hypothesis, okay? If, and then. The if part of our hypothesis needs to talk about how plants transport water. Okay, so we've talked about that, the whole osmotic pulling action, where it comes from, where it starts, why it happens, polar nature of water, okay, all that kind of stuff, okay? Okay, and I don't expect like a paragraph here, okay? It's not just simple, okay? Give me the basics about what forces are involved in making water go up a plant. Okay, you don't need to go into the molecular biology, okay, of the inside of the xylem and its nature and cellulose and all that. You don't need to. Okay, it doesn't need to be that detailed. Just talk about the forces that are there. Okay, and part, as always, describes the experiment. Probably an important detail here would be how we changed the plant. Okay, it would be important to note that there's only a leaf here. Right? The rest of the plant is no longer there. Okay? That helps us to isolate the source of this transporting force. Okay? The then part is simple. Predict your results. There's two things here you should talk about, and they're your two responding variables. How are they going to change? Okay, talk about what's going to be different with your experimental setup on day 2 or day 3, okay, versus day 1 or day 0 and day 2, whichever way makes more sense to you. Okay, what's going to be different? Okay, that's your prediction, because if plants transport water the way you think they do, and you do this, then what are you going to see? Okay? That'll be an easy hypothesis to accept or reject when you're done. Okay? Either it's going to do it, or it's not. Okay, so we did all the procedure stuff yesterday. Okay, hopefully everybody made that one really important observation, that one important quantitative observation yesterday, and that was your initial volume of water that's in your graduated cylinder. Okay, that's why I kept saying when we were in the lab, make sure you pour your water in there, and then you make a note of how much is left in the big cylinder. Okay, subtract that from 100. That's what's in your graduated cylinder. I sure hope everybody did that, or we're pretty screwed in terms of your water volume observations. Okay, so make sure you've got that observation. That's going to be an important observation. So starting volume and ending volume, those are going to be your two observations, or your observations there. And then obviously you've been taking pictures of the plant. You took them on day 1, you took them again today, you're going to take them again tomorrow. Okay, hopefully you see a bit more of a change in those plants tomorrow. It's supposed to be pretty sunny today. Okay. All right, then for our analysis and interpretation, obviously we can't do that yet because we don't have all of our observations, okay? But the first one is really simple. All you have to do is calculate the amount of water your plant took up. So that would be initial volume minus final volume, and there's the amount of water that your plant took up. Okay, typically when we do this lab, which obviously this time we're doing with a different type of plant because they didn't have the Dieffenbachia we usually use, okay? Typically, we get like 5 mils difference, okay? Between 3 and 6 usually, okay? Over the 2 days. Hopefully that's what we're going to see here as well, okay? So that'll be your first question, basically just calculating that and then talking about it a little bit, okay? Question number 2, what evidence do you have that water followed the path that we discussed in class? That would be up the xylem into the spongy layer and evaporating out through what structures? The stomates. Okay, remember the stomates are those things that open and close, okay, based on how much salt and how much water the plant has in it. Okay, water evaporates through there. Since we colored the water, hopefully we'll be able to trace its path through the plant. We'll be able to actually see it, okay, as it moves through the plant. What you'll be looking for tomorrow is if you see little dyed dots on the bottom of your leaf, that's where the stomates are, because the dye can't evaporate. The water can, and the dye gets left behind, And where it gets left behind is right where the water evaporates at the stomate. So hopefully we'll be able to see some little colored dots on the bottom of your leaves tomorrow. Okay, so you got to talk a bit about that. All right, and then number 3, this is the one where you have to think about it a little bit. Okay, think about how osmosis works. Okay, think about how salts get left behind in the leaf. And then predict the effect of putting those, instead of in tap water, in salt water. Okay, what would happen then? Would this experiment have the same results? Would they be different? How would they be different? Okay, that's what you have to do for number 3. Okay, based on what you know of how a plant transports water, how would this work differently if I put salt water there instead of fresh water? Right, and then same as always, conclusion and evaluation. Okay, sources of error, okay, anything like that. When you're thinking of sources of error, okay, most of them are going to be conditional. Okay, think about what kinds of things might change in this room over the 48-hour period of the experiment. Okay, keeping in mind that the lights in this room are on sensors. So once there's no one walking around in here for about 10 minutes, the lights turn off. Okay. The temperature, however, is thermostatically controlled. It's right over there. And I won't touch it for 2 days. Okay. All right. Questions on what we're doing then? Okay, so I'm going to give you guys, let's say about 20 minutes. You can work on that lab report a little bit, get your design, hypothesis, stuff like that kind of set up. Okay, because I usually give you some time for the pre-lab, and we didn't get a chance to do that yesterday. So I'm going to give you about 20 minutes or so here, okay, to work on that lab report. We'll see how much of it you can get done in class, and because tomorrow we won't have